Photo shows a mottled brown chameleon that blends into the leaf it sits on. Figure 27. 1. The leaf chameleon, Brachysia micra, was discovered in northern Madagascar in 2012. At just over 1 inch long, it is the smallest known chameleon. Credit: Modification of work by Frank Glaw, et al., PLOS. Chapter outline: 27. 1 Features of the animal kingdom. 27. 2 Features used to classify animals. 27. 3 Animal phylogeny. 27. 4 The evolutionary history of the animal kingdom. Animal evolution began in the ocean over 600 million years ago with tiny creatures that probably do not resemble any living organism today. Since then, animals have evolved into a highly diverse kingdom. Although over 1 million extant, currently living, species of animals have been identified, scientists are continually discovering more species as they explore ecosystems around the world. The number of extant species is estimated to be between 3 and 30 million. But what is an animal? While we can easily identify dogs, birds, fish, spiders, and worms as animals, other organisms, such as corals and sponges, are not as easy to classify. Animals vary in complexity, from sea sponges to crickets to chimpanzees, and scientists are faced with the difficult task of classifying them within a unified system. They must identify traits that are common to all animals as well as traits that can be used to distinguish among related groups of animals. The animal classification system characterizes animals based on their anatomy, morphology, evolutionary history, features of embryological development, and genetic makeup. This classification scheme is constantly developing as new information about species arises. Understanding and classifying the great variety of living species help us better understand how to conserve the diversity of life on Earth. Learning objectives. By the end of this section, you will be able to do the following. List the features that distinguish the kingdom Animalia from other kingdoms. Explain the processes of animal reproduction and embryonic development. Describe the roles that Hox genes play in development. 2 different groups within the domain Eukaryota have produced complex multicellular organisms. The plants arose within the Archaeplastida, whereas the animals and their close relatives, the fungi, arose within the Opisthokonta. However, plants and animals not only have different lifestyles, they also have different cellular histories as eukaryotes. The Opisthokonts share the possession of a single posterior flagellum in flagellated cells, e.g., sperm cells. Most animals also share other features that distinguish them from organisms in other kingdoms. All animals require a source of food and are therefore heterotrophic, ingesting other living or dead organisms. This feature distinguishes them from autotrophic organisms, such as most plants, which synthesize their own nutrients through photosynthesis. As heterotrophs, animals may be carnivores, herbivores, omnivores, or parasites. Figure 27. 2a-b. As with plants, almost all animals have a complex tissue structure with differentiated and specialized tissues. The necessity to collect food has made most animals motile, at least during certain life stages. The typical life cycle in animals is diplontic. Like you, the diploid state is multicellular, whereas the haploid state is gametic, such as sperm or egg. We should note that the alternation of generations characteristic of the land plants is typically not found in animals. In animals whose life histories include several to multiple body forms, e.g., insect larvae or the medusae of some cnidarians, all body forms are diploid. Animal embryos pass through a series of developmental stages that establish a determined and fixed body plan. The body plan refers to the morphology of an animal determined by developmental cues. Part A shows a bear with a large fish in its mouth. Part B shows a heart in a jar. Long, thread-like worms extend from the heart. Figure 27. 2 Heterotrophy. All animals are heterotrophs and thus derive energy from a variety of food sources. The black bear is an omnivore, eating both plants and animals. The B, heartworm Dirofilaria amidus, is a parasite that derives energy from its hosts. It spends its larval stage in mosquitoes and its adult stage infesting the heart of dogs and other mammals, as shown here. Credit A. Modification of work by USDA Forest Service. Credit B. Modification of work by Clyde Robinson. Complex tissue structure. Many of the specialized tissues of animals, are associated with the requirements and hazards of seeking and processing food. This explains why animals typically have evolved special structures associated with specific methods of food capture and complex digestive systems supported by accessory organs. Sensory structures help animals navigate their environment, detect food sources, and avoid becoming a food source for other animals. Movement is driven by muscle tissue attached to supportive structures like bone or chitin and is coordinated by neural communication. Animal cells may also have unique structures for intercellular communication, such as gap junctions. The evolution of nerve tissues and muscle tissues has resulted in animals' unique ability to rapidly sense and respond to changes in their environment. This allows animals to survive in environments where they must compete with other species to meet their nutritional demands. The tissues of animals differ from those of the other major multicellular eukaryotes, plants, and fungi, because their cells don't have cell walls. However, cells of animal tissues may be embedded in an extracellular matrix. E.g., mature bone cells reside within a mineralized organic matrix secreted by the cells. In vertebrates, bone tissue is a type of connective tissue that supports the entire body structure. The complex bodies and activities of vertebrates demand such supportive tissues. Epithelial tissues cover and protect both external and internal body surfaces and may also have secretory functions. Epithelial tissues include the epidermis of the integument, the lining of the digestive tract and trachea, as well as the layers of cells that make up the ducts of the liver and glands of advanced animals, for example. The different types of tissues in true animals are responsible for carrying out specific functions for the organism. This differentiation and specialization of tissues is part of what allows for such incredible animal diversity. Just as there are multiple ways to be a eukaryote, there are multiple ways to be a multicellular animal. The animal kingdom is currently divided into 5 monophyletic clades: Parazoa or porifera, sponges; Placozoa, tiny parasitic creatures that resemble multicellular amoebae; Cnidaria, jellyfish and their relatives; Ctenophora, the comb jellies; and Bilateria, all other animals. The Placozoa, flat animal, and Parazoa, beside animal, do not have specialized tissues derived from germ layers of the embryo, although they do possess specialized cells that act functionally like tissues. The Placozoa have only 4 cell types, while the sponges have nearly 24. The 3 other clades do include animals with specialized tissues derived from the germ layers of the embryo. In spite of their superficial similarity to Cnidarian medusae, recent molecular studies indicate that the ctenophores are only distantly related to the cnidarians, which together with the Bilateria constitute the Eumetozoa, true animals. When we think of animals, we usually think of Eumetozoa since most animals fall into this category. Link to learning. Watch a presentation by biologist E.O. Wilson on the importance of diversity. Animal reproduction and development. Most animals are diploid organisms, meaning that their body somatic cells are diploid and haploid reproductive gamete cells are produced through meiosis. Some exceptions exist. For example, in bees, wasps, and ants, the male is haploid because it develops from unfertilized eggs. Most animals undergo sexual reproduction. However, a few groups, such as cnidarians, flatworms, and roundworms, may also undergo asexual reproduction, in which offspring originate from part of the parental body. Processes of animal reproduction and embryonic development. During sexual reproduction, the haploid gametes of the male and female individuals of a species combine in a process called fertilization. Typically, both male and female gametes are required. The small, motile male sperm fertilizes the typically much larger, sessile female egg. This process produces a diploid fertilized egg called a zygote. Some animal species, including sea stars and sea anemones, are capable of asexual reproduction. The most common forms of asexual reproduction for stationary aquatic animals include budding and fragmentation, where part of a parent individual can separate and grow into a new individual. This type of asexual reproduction produces genetically identical offspring, which would appear to be disadvantageous from the perspective of evolutionary adaptability, simply because of the potential buildup of deleterious mutations. In contrast, a form of uniparental reproduction found in some insects and a few vertebrates is called parthenogenesis, or virgin beginning. In this case, progeny develop from a gamete, but without fertilization. Because of the nutrients stored in eggs, only females produce parthenogenetic offspring. In some insects, unfertilized eggs develop into new male offspring. This type of sex determination is called haplodiploidy, since females are diploid with both maternal and paternal chromosomes, and males are haploid with only maternal chromosomes. A few vertebrates, e.g., some fish, turkeys, rattlesnakes, and whiptail lizards, are also capable of parthenogenesis. In the case of turkeys and rattlesnakes, parthenogenetically reproducing females also produce only male offspring, but not because the males are haploid. In birds and rattlesnakes, the female is the heterogametic ZW sex, so the only surviving progeny of postmeiotic parthenogenesis would be ZZ males. In the whiptail lizards, on the other hand, only female progeny are produced by parthenogenesis. These animals may not be identical to their parent, although they have only maternal chromosomes. However, for animals that are limited in their access to mates, uniparental reproduction can ensure genetic propagation. In animals, the zygote progresses through a series of developmental stages, during which primary germ layers, ectoderm, endoderm, and mesoderm, are established and reorganized to form an embryo. During this process, animal tissues begin to specialize and organize into organs and organ systems, determining their future morphology and physiology. Animal development begins with cleavage, a series of mitotic cell divisions, of the zygote, figure 27. 3. Cleavage differs from somatic cell division in that the egg is subdivided by successive cleavages into smaller and smaller cells, with no actual cell growth. The cells resulting from subdivision of the material of the egg in this way are called blastomeres. 3 cell divisions transform the single-celled zygote into an 8-celled structure. After further cell division and rearrangement of existing cells, a solid morula is formed, followed by a hollow structure called a blastula. The blastula is hollow only in invertebrates whose eggs have relatively small amounts of yolk. In very yolky eggs of vertebrates, the yolk remains undivided, with most cells forming an embryonic layer on the surface of the yolk. Imagine a chicken embryo growing over the egg's yolk, which serve as food for the developing embryo. Further cell division and cellular rearrangement leads to a process called gastrulation. Gastrulation results in 2 important events: the formation of the primitive gut, archenteron, or digestive cavity, and the formation of the embryonic germ layers, as we have discussed above. These germ layers are programmed to develop into certain tissue types, organs, and organ systems during a process called organogenesis. Diploblastic organisms have 2 germ layers, endoderm and ectoderm. Endoderm forms the wall of the digestive tract, and ectoderm covers the surface of the animal. In triploblastic animals, a 3rd layer forms, mesoderm, which differentiates into various structures between the ectoderm and endoderm, including the lining of the body cavity. The left part of the illustration shows a single-celled zygote. The initial cleavage, or cell division, results in a ball of cells, called the 8-cell stage. The cells do not grow during cleavage, so the 8-cell stage ball is about the same diameter as the zygote. Further cleavage results in a hollow ball of cells called a blastula. Upon gastrulation, part of the ball of cells invaginates, forming a cavity called a blastopore. Figure 27, 3 development of a simple embryo. During embryonic development, the zygote undergoes a series of mitotic cell divisions, or cleavages, that subdivide the egg into smaller and smaller blastomeres. Note that the 8-cell stage and the blastula are about the same size as the original zygote. In many invertebrates, the blastula consists of a single layer of cells around a hollow space. During a process called gastrulation, the cells from the blastula move inward on one side to form an inner cavity. This inner cavity becomes the primitive gut, archenteron, of the gastrula, little gut, stage. The opening into this cavity is called the blastopore, and in some invertebrates it is destined to form the mouth. Some animals produce larval forms that are different from the adult. In insects with incomplete metamorphosis, such as grasshoppers, the young resemble wingless adults, but gradually produce larger and larger wing buds during successive molts, until finally producing functional wings and sex organs during the last molt. Other animals, such as some insects and echinoderms, undergo complete metamorphosis in which the embryo develops into one or more feeding larval stages that may differ greatly in structure and function from the adult, figure 27. 4. The adult body then develops from one or more regions of larval tissue. For animals with complete metamorphosis, the larva and the adult may have different diets, limiting competition for food between them. Regardless of whether a species undergoes complete or incomplete metamorphosis, the series of developmental stages of the embryo remains largely the same for most members of the animal kingdom. Illustration A shows the egg, nymph, and adult stages of a grasshopper. The nymph stages are similar in appearance to the adult stage, but smaller. Illustration B shows the egg, larvae, pupa, and adult stages of a butterfly. The pupa is a cocoon the butterfly makes when transforming from the larval to adult stages. The winged adult butterfly looks nothing like the caterpillar larva. Figure 27. 4. Insect metamorphosis. A. The grasshopper undergoes incomplete metamorphosis. B. The butterfly undergoes complete metamorphosis. Credit, S.E. Snodgrass, USDA. Link to learning. Watch the following video to see how human embryonic development after the blastula and gastrula stages of development reflects evolution. The role of homeobox, HOX, genes in animal development. Since the early 19th century, scientists have observed that many animals, from the very simple to the complex, shared similar embryonic morphology and development. Surprisingly, a human embryo and a frog embryo, at a certain stage of embryonic development, look remarkably alike. For a long time, scientists did not understand why so many animal species looked similar during embryonic development, but were very different as adults. They wondered what dictated the developmental direction that a fly, mouse, frog, or human embryo would take. Near the end of the 20th century, a particular class of genes was discovered that had this very job. These genes that determine animal structure are called homeotic genes, and they contain DNA sequences called homeoboxes. The work to understand the role these genes play was novel and incredibly detailed. Over several years, Christiane Nusselen-Volhard and Eric Wieshaus introduced mutated genes into fruit flies and observed changes to the flies' bodies under microscopes. Eventually, they were able to identify specific changes, such as different numbers of body segments, based on mutations of specific genes, therefore showing which genes controlled aspects of development. Genes with homeoboxes encode protein transcription factors. One group of animal genes containing homeobox sequences is specifically referred to as HOX genes. This cluster of genes is responsible for determining the general body plan, such as the number of body segments of an animal, the number and placement of appendages, and animal head-tail directionality. The first HOX genes to be sequenced were those from the fruit fly, Drosophila melanogaster. A single HOX mutation in the fruit fly can result in an extra pair of wings or even legs growing from the head in place of antennae. This is because antennae and legs are embryologic homologous structures, and their appearance as antennae or legs is dictated by their origination within specific body segments of the head and thorax during development. Now, HOX genes are known from virtually all other animals as well. While there are a great many genes that play roles in the morphological development of an animal, including other homeobox-containing genes, what makes HOX genes so powerful is that they serve as master control genes that can turn on or off large numbers of other genes. HOX genes do this by encoding transcription factors that control the expression of numerous other genes. HOX genes are homologous across the animal kingdom. That is, the genetic sequences of HOX genes and their positions on chromosomes are remarkably similar across most animals because of their presence in a common ancestor, from worms to flies, mice, and humans, figure 27. 5. In addition, the order of the genes reflects the anterior-posterior axis of the animal's body. One of the contributions to increased animal body complexity is that HOX genes have undergone at least 2, and perhaps as many as 4 duplication events during animal evolution, with the additional genes allowing for more complex body types to evolve. All vertebrates have 4 or more sets of Hox genes, while invertebrates have only one set. Visual connection. This illustration shows the 4 clusters of Hox genes found in vertebrates. Hox A, Hox B, Hox C, and Hox D. There are 13 Hox genes, but not all of them are found in each cluster. In both mice and humans, genes 1 through 4 regulate the development of the head. Genes 5 and 6 regulate the development of the neck. Genes 7 and 8 regulate the development of the torso, and genes 9 through 13 regulate the development of the arms and legs. Figure 27. 5 Hox genes. Hox genes are highly conserved genes encoding transcription factors that determine the course of embryonic development in animals. In vertebrates, the genes have been duplicated into 4 clusters on different chromosomes. Hox A, Hox B, Hox C, and Hox D genes within these clusters are expressed in certain body segments at certain stages of development. Shown here is the homology between Hox genes in mice and humans. Note how Hox gene expression, as indicated with orange, pink, blue, and green shading, occurs in the same body segments in both the mouse and the human. While at least one copy of each Hox gene is present in humans and other vertebrates, some Hox genes are missing in some chromosomal sets. If a Hox13 gene in a mouse was replaced with a Hox1 gene, how might this alter animal development? 2 of the 5 clades within the animal kingdom do not have Hox genes, the Ctenophora and the Porifera. In spite of the superficial similarities between the Cnidaria and the Ctenophora, the Cnidaria have a number of Hox genes, but the Ctenophora have none. The absence of Hox genes from the ctenophores has led to the suggestion that they might be basal animals, in spite of their tissue differentiation. Ironically, the Placozoa, which have only a few cell types, do have at least one Hox gene. The presence of a Hox gene in the Placozoa, in addition to similarities in the genomic organization of the Placozoa, Cnidaria, and Bilateria, has led to the inclusion of the 3 groups in a Parahoxozoa clade. However, we should note that at this time, the reclassification of the animal kingdom is still tentative and requires much more study. Learning objectives. By the end of this section, you will be able to do the following. Explain the differences in animal body plans that support basic animal classification. Compare and contrast the embryonic development of protostomes and deuterostomes. Scientists have developed a classification scheme that categorizes all members of the animal kingdom. Although there are exceptions to most rules governing animal classification, Figure 27. 6. Animals have been traditionally classified according to 2 characteristics: body plan and developmental pathway. The major feature of the body plan is its symmetry, how the body parts are distributed along the major body axis. Symmetrical animals can be divided into roughly equivalent halves along at least one axis. Developmental characteristics include the number of germ tissue layers formed during development, the origin of the mouth and anus, the presence or absence of an internal body cavity, and other features of embryological development, such as larval types or whether or not periods of growth are interspersed with molting. Visual connection. The phylogenetic tree of metazoans, or animals, branches into parazoans with no tissues, sponges together with Ctenophora as sister branches, and Parahoxozoa, which branches into Placozoa and Eumetazoans with specialized tissues. Eumetazoans branch into Radiata, diploblastic animals with radial symmetry, and Bilateria, triploblastic animals with bilateral symmetry. Radiata includes cnidarians. Bilateria branches into Acula, which have no body cavity, and Protostomia and Deuterostomia, which possess a body cavity. Deuterostomes include chordates and echinoderms. Protostomia branches into Lophotrochozoa and Ecdysozoa. Ecdysozoa includes arthropods and nematodes, or roundworms. Lophotrochozoa includes mollusca, annelida, brachiopoda, ectoprocta, rotifera, and platyhelminthes. Figure 27. 6. Animal phylogeny. The phylogenetic tree of animals is based on morphological, fossil, and genetic evidence. The Ctenophora and Porifera are both considered to be basal because of the absence of Hox genes in this group, but how they are related to the Parahoxozoa, Placozoa plus Eumetazoa, or to each other, continues to be a matter of debate. The represented tree is one of several proposed relationships based on genetic and other data. Which of the following statements is false? Eumetozoans have specialized tissues and parazoans don't. Lophotrochozoa and ecdysozoa are both bilateria. Aquila and Cnidaria both possess radial symmetry. Arthropods are more closely related to nematodes than they are to annelids. Animal characterization based on body symmetry. At a very basic level of classification, true animals can be largely divided into 3 groups based on the type of symmetry of their body plan: radially symmetrical, bilaterally symmetrical, and asymmetrical. Asymmetry is seen in 2 modern clades, the Parazoa, figure 27, 7a, and Placozoa. Although we should note that the ancestral fossils of the Parazoa apparently exhibited bilateral symmetry. One clade, the Cnidaria, figure 27, 7b, c, exhibits radial or biradial symmetry. Ctenophores have rotational symmetry, figure 27, 7e. Bilateral symmetry is seen in the largest of the clades, the Bilateria, figure 27, 7d. However, the Echinodermata are bilateral as larvae and metamorphose secondarily into radial adults. All types of symmetry are well suited to meet the unique demands of a particular animal's lifestyle. Radial symmetry is the arrangement of body parts around a central axis, as is seen in a bicycle wheel or pie. It results in animals having top and bottom surfaces, but no left and right sides, nor front or back. If a radially symmetrical animal is divided in any direction along the oral-aboral axis, the side with a mouth is oral side, and the side without a mouth is the aboral side. The 2 halves will be mirror images. This form of symmetry marks the body plans of many animals in the phyla Cnidaria, including jellyfish and adult sea anemones, figure 27. 7b, c. Radial symmetry equips these sea creatures, which may be sedentary or only capable of slow movement or floating, to experience the environment equally from all directions. Bilaterally symmetrical animals, like butterflies, figure 27, 7d, have only a single plane along which the body can be divided into equivalent halves. The Ctenophora, figure 27, 7e, although they look similar to jellyfish, are considered to have rotational symmetry rather than radial or biradial symmetry, because division of the body into 2 halves along the oral-aboral axis divides them into 2 copies of the same half, with one copy rotated 180°, rather than 2 mirror images. Part A shows several sponges, which form irregular, bumpy blobs on the seafloor. Part B shows a jellyfish with long, slender tentacles radiating from a flexible, disc-shaped body. Part C shows an anemone sitting on the seafloor with thick tentacles radiating up from a cup-shaped body. Part D shows a black butterfly with 2 symmetrical wings. Part E shows a burrow, which is a type of jellyfish, semi-transparent with more solid ribs and a visible opening at one end. Figure 27, 7 symmetry in animals. The A sponge is asymmetrical. The B jellyfish and C anemone are radially symmetrical. The D butterfly is bilaterally symmetrical. Rotational symmetry, E, is seen in the ctenophore burrow, shown swimming open-mouthed. Credit A. Modification of work by Andrew Turner. Credit B. Modification of work by Robert Freiberger. Credit C. Modification of work by Samuel Chow. Credit D. Modification of work by Corey Zanker. Credit E. Modification of work by NOAA. Bilateral symmetry involves the division of the animal through a mid-sagittal plane, resulting in 2 superficially mirror images, right and left halves, such as those of a butterfly. Figure 27. 7d, crab or human body. Animals with bilateral symmetry have a head and tail, anterior V's posterior, front and back dorsal V's ventral, and right and left sides, figure 27. 8. All eumetozoa except those with secondary radial symmetry are bilaterally symmetrical. The evolution of bilateral symmetry that allowed for the formation of anterior and posterior, head and tail, ends promoted a phenomenon called cephalization, which refers to the collection of an organized nervous system at the animal's anterior end. In contrast to radial symmetry, which is best suited for stationary or limited motion lifestyles, bilateral symmetry allows for streamlined and directional motion. In evolutionary terms, this simple form of symmetry promoted active and controlled directional mobility and increased sophistication of resource-seeking and predator-prey relationships. The illustration shows a woman's body dissected into planes. The coronal plane separates the front from the back. The front of the body is the ventral side, and the back of the body is the dorsal side. The transverse plane separates the upper body from the lower body, running parallel to the ground. The upper body is defined as cranial, and the lower body is defined as caudal. The sagittal plane dissects the body from side to side. The medial line goes through the center of the body. The areas to the left and right of the medial line are defined as lateral. Parts of the body close to the medial line are proximal, and those further away are distal. Figure 27, 8 bilateral symmetry. The bilaterally symmetrical human body can be divided by several planes. Animals in the phylum Echinodermata, such as sea stars, sand dollars, and sea urchins, display modified radial symmetry as adults. But as we have noted, their larval stages, such as the Bipinnaria, initially exhibit bilateral symmetry until they metamorphose in animals with radial symmetry. This is termed secondary radial symmetry. Echinoderms evolved from bilaterally symmetrical animals, thus they are classified as bilaterally symmetrical. Link to learning. Watch this video to see a quick sketch of the different types of body symmetry. Animal characterization, based on features of embryological development. Most animal species undergo a separation of tissues into germ layers during embryonic development. Recall that these germ layers are formed during gastrulation, and that each germ layer typically gives rise to specific types of embryonic tissues and organs. Animals develop either 2 or 3 embryonic germ layers. Figure 27. 9. The animals that display radial, biradial, or rotational symmetry develop 2 germ layers, an inner layer, endoderm or mesendoderm, and an outer layer, ectoderm. These animals are called diploblasts and have a non-living middle layer between the endoderm and ectoderm. Although individual cells may be distributed through this middle layer, there is no coherent third layer of tissue. The 4 clades considered to be diploblastic have different levels of complexity and different developmental pathways, although there is little information about development in Placozoa. More complex animals, usually those with bilateral symmetry, develop 3 tissue layers, an inner layer endoderm, an outer layer ectoderm, and a middle layer mesoderm. Animals with 3 tissue layers are called triploblasts. Visual connection. The left illustration shows the 2 embryonic germ layers of a diploblast. The inner layer is the endoderm, and the outer layer is the ectoderm. Sandwiched between the endoderm and the ectoderm is a non-living layer. Right illustration shows the 3 embryonic germ layers of a triploblast. Like the diploblast, the triploblast has an inner endoderm and an outer ectoderm. Sandwiched between these 2 layers is a living mesoderm. Figure 27. 9 diploblastic and triploblastic embryos. During embryogenesis, diploblasts develop 2 embryonic germ layers, an ectoderm and an endoderm or mesendoderm. Triploblasts develop a 3rd layer, the mesoderm, which arises from mesendoderm and resides between the endoderm and ectoderm. Which of the following statements about diploblasts and triploblasts is false? Animals that display only radial symmetry during their lifespans are diploblasts. Animals that display bilateral symmetry are triploblasts. The endoderm gives rise to the lining of the digestive tract and the respiratory tract. The mesoderm gives rise to the central nervous system. Each of the 3 germ layers is programmed to give rise to specific body tissues and organs, although there are variations on these themes. Generally speaking, the endoderm gives rise to the lining of the digestive tract, including the stomach, intestines, liver, and pancreas, as well as to the lining of the trachea, bronchi, and lungs of the respiratory tract, along with a few other structures. The ectoderm develops into the outer epithelial covering of the body surface, the central nervous system, and a few other structures. The mesoderm is the 3rd germ layer. It forms between the endoderm and ectoderm in triploblasts. This germ layer gives rise to all specialized muscle tissues, including the cardiac tissues and muscles of the intestines, connective tissues such as the skeleton and blood cells, and most other visceral organs such as the kidneys and the spleen. Diploblastic animals may have cell types that serve multiple functions, such as epitheliomuscular cells, which serve as a covering as well as contractile cells. Presence or absence of a coelom. Further subdivision of animals with 3 germ layers, triploblasts, results in the separation of animals that may develop an internal body cavity derived from mesoderm, called a coelom, and those that do not. This epithelial cell-lined coelomic cavity, usually filled with fluid, lies between the visceral organs and the body wall. It houses many organs such as the digestive, urinary, and reproductive systems, the heart and lungs, and also contains the major arteries and veins of the circulatory system. In mammals, the body cavity is divided into the thoracic cavity, which houses the heart and lungs, and the abdominal cavity, which houses the digestive organs. In the thoracic cavity, further subdivision produces the pleural cavity, which provides space for the lungs to expand during breathing, and the pericardial cavity, which provides room for movements of the heart. The evolution of the coelom is associated with many functional advantages. For example, the coelom provides cushioning and shock absorption for the major organ systems that it encloses. In addition, organs housed within the coelom can grow and move freely, which promotes optimal organ development and placement. The coelom also provides space for the diffusion of gases and nutrients, as well as body flexibility, promoting improved animal motility. Triploblasts that do not develop a coelom are called achylomates, and their mesoderm region is completely filled with tissue, although they do still have a gut cavity. Examples of achylomates include animals in the phylum Platyhelminthes, also known as flatworms. Animals with a true coelom are called eucholomates or coelomates, figure 27:10. In such cases, a true coelom arises entirely within the mesoderm germ layer and is lined by an epithelial membrane. This membrane also lines the organs within the coelom, connecting and holding them in position while allowing them some freedom of movement. Annelids, mollusks, arthropods, echinoderms, and chordates are all eucholomates. A third group of triploblasts has a slightly different coelom lined partly by mesoderm and partly by endoderm. Although still functionally a coelom, these are considered false coeloms, and so we call these animals pseudocoelomates. The phylum Nematoda, roundworms, is an example of a pseudocoelomate. True coelomates can be further characterized based on other features of their early embryological development. Part A shows the body plan of achylomates, including flatworms. Achylomates have a central digestive cavity. Outside this digestive cavity are 3 tissue layers: an inner endoderm, a central mesoderm, and an outer ectoderm. The photo shows a swimming flatworm, which has the appearance of a frilly black and pink ribbon. Part B shows the body plan of eucholomates, which include annelids, mollusks, arthropods, echinoderms, and chordates. Eucholomates have the same tissue layers as achylomates, but a cavity called a coelom exists within the mesoderm. The coelom is divided into 2 symmetrical parts that are separated by 2 spokes of mesoderm. The photo shows a swimming annelid known as a bloodworm. The bloodworm has a tubular body that tapers at each end. Numerous appendages radiate from either side. Part C shows the body plan of pseudocoelomates, which include roundworms. Like the achylomates and eucholomates, the pseudocoelomates have an endoderm, a mesoderm, and an ectoderm. However, in pseudocoelomates, a pseudocoelom separates the endoderm from the mesoderm. The photo shows a roundworm or nematode, which has a tubular body. Figure 27, 10 body cavities. Triploblasts may be A. achylomates, B. eucholomates, or C. pseudocoelomates. Achylomates have no body cavity. Eucholomates have a body cavity within the mesoderm, called a coelom, in which both the gut and the body wall are lined with mesoderm. Pseudocoelomates also have a body cavity, but only the body wall is lined with mesoderm. Credit, a modification of work by Jan Dirk. Credit B, modification of work by NOAA. Credit C, modification of work by USDA, ARS. Embryonic development of the mouth. Bilaterally symmetrical, triploblastic eucholomates can be further divided into 2 groups based on differences in the origin of the mouth. When the primitive gut forms, the opening that first connects the gut cavity to the outside of the embryo is called the blastopore. Most animals have openings at both ends of the gut, mouth at one end and anus at the other. One of these openings will develop at or near the site of the blastopore. In protostomes, mouth first, the mouth develops at the blastopore, figure 27:11. In deuterostomes, mouth second, the mouth develops at the other end of the gut, figure 2711, and the anus develops at the site of the blastopore. Protostomes include arthropods, mollusks, and annelids. Deuterostomes include more complex animals such as chordates, but also some simple animals such as echinoderms. Recent evidence has challenged this simple view of the relationship between the location of the blastopore and the formation of the mouth, however, and the theory remains under debate. Nevertheless, these details of mouth and anus formation reflect general differences in the organization of protostome and deuterostome embryos, which are also expressed in other developmental features. One of these differences between protostomes and deuterostomes is the method of coelom formation, beginning from the gastrula stage. Since body cavity formation tends to accompany the formation of the mesoderm of protostomes and deuterostomes forms differently. The coelom of most protostomes is formed through a process called schizocoly. The mesoderm in these organisms is usually the product of specific blastomeres, which migrate into the interior of the embryo and form two clumps of mesodermal tissue. Within each clump, cavities develop and merge to form the hollow opening of the coelom. Deuterostomes differ in that their coelom forms through a process called enterocoely. Here, the mesoderm develops as pouches that are pinched off from the endoderm tissue. These pouches eventually fuse and expand to fill the space between the gut and the body wall, giving rise to the coelom. Another difference in organization of protostome and deuterostome embryos is expressed during cleavage. Protostomes undergo spiral cleavage, meaning that the cells of one pole of the embryo are rotated and thus misaligned with respect to the cells of the opposite pole. This is due to the oblique angle of cleavage relative to the two poles of the embryo. Deuterostomes undergo radial cleavage, where the cleavage axes are either parallel or perpendicular to the polar axis, resulting in the parallel, up and down alignment of the cells between the two poles. The illustration compares the development of protostomes and deuterostomes. In both protostomes and deuterostomes, the gastrula, which resembles a hollow ball of cells, contains an indentation called a blastopore. In protostomes, two circular layers of mesoderm form inside the gastrula, containing the coelom cavity. As the protostome develops, the mesoderm grows and fuses with the gastrula cell layer. The blastopore becomes the mouth, and a second opening forms opposite the mouth, which becomes the anus. In deuterostomes, two groups of gastrula cells in the blastopore grow inward to form the mesoderm. As the deuterostome develops, the mesoderm pinches off and fuses, forming a second body cavity. The body plan of the deuterostome at this stage looks very similar to that of the protostome, but the blastopore becomes the anus, and the second opening becomes the mouth. Figure 27. 11 protostomes and deuterostomes. Eucholomates can be divided into two groups based on their early embryonic development. In protostomes, the mouth forms at or near the site of the blastopore, and the body cavity forms by splitting the mesodermal mass during the process of schizocoly. In deuterostomes, the mouth forms at a site opposite the blastopore end of the embryo, and the mesoderm pinches off to form the coelom during the process of enterocoely. A second distinction between the types of cleavage in protostomes and deuterostomes relates to the fate of the resultant blastomeres, cells produced by cleavage. In addition to spiral cleavage, protostomes also undergo determinate cleavage. This means that even at this early stage, the developmental fate of each embryonic cell is already determined. A given cell does not have the ability to develop into any cell type other than its original destination. Removal of a blastomere from an embryo with determinate cleavage can result in missing structures and embryos that fail to develop. In contrast, deuterostomes undergo indeterminate cleavage, in which cells are not yet fully committed at this early stage to develop into specific cell types. Removal of individual blastomeres from these embryos does not result in the loss of embryonic structures. In fact, twins clones can be produced as a result from blastomeres that have been separated from the original mass of blastomere cells. Unlike protostomes, however, if some blastomeres are damaged during embryogenesis, adjacent cells are able to compensate for the missing cells, and the embryo is not damaged. These cells are referred to as undetermined cells. This characteristic of deuterostomes is reflected in the existence of familiar embryonic stem cells, which have the ability to develop into any cell type until their fate is programmed at a later developmental stage. Evolution connection. The evolution of the coelom. One of the first steps in the classification of animals is to examine the animal's body. One structure that is used in classification of animals is the body cavity or coelom. The body cavity develops within the mesoderm, so only triploblastic animals can have body cavities. Therefore, body cavities are found only within the bilateria. In other animal clades, the gut is either close to the body wall or separated from it by a jelly-like material. The body cavity is important for two reasons. Fluid within the body cavity protects the organs from shock and compression. In addition, since in triploblastic embryos, most muscle, connective tissue, and blood vessels develop from mesoderm, these tissues developing within the lining of the body cavity can reinforce the gut and body wall, aid in motility, and efficiently circulate nutrients. To recap what we have discussed above, animals that do not have a coelom are called acoelomates. The major acoelomate group in the bilateria is the flatworms, including both free-living and parasitic forms such as tapeworms. In these animals, mesenchyme fills the space between the gut and the body wall. Although two layers of muscle are found just under the epidermis, there is no muscle or other mesodermal tissue around the gut. Flatworms rely on passive diffusion for nutrient transport across their body. In pseudocoelomates, there is a body cavity between the gut and the body wall, but only the body wall has mesodermal tissue. In these animals, the mesoderm forms but does not develop cavities within it. Major pseudocoelomate phyla are the rotifers and nematodes. Animals that have a true coelom are called eucoelomates. All vertebrates, as well as mollusks, annelids, arthropods, and echinoderms, are eucoelomates. The coelom develops within the mesoderm during embryogenesis. Of the major bilaterian phyla, the mollusks, annelids, and arthropods are schizocoels, in which the mesoderm splits to form the body cavity, while the echinoderms and chordates are enterocoels, in which the mesoderm forms as two or more buds off of the gut. These buds separate from the gut and coalesce to form the body cavity. In the vertebrates, mammals have a subdivided body cavity, with the thoracic cavity separated from the abdominal cavity. The pseudocoelomates may have had eucoelomate ancestors and may have lost their ability to form a complete coelom through genetic mutations. Thus, this step in early embryogenesis, the formation of the coelom, has had a large evolutionary impact on the various species of the animal kingdom. Learning objectives. By the end of this section, you will be able to do the following. Interpret the metazoan phylogenetic tree. Describe the types of data that scientists use to construct and revise animal phylogeny. List some of the relationships within the modern phylogenetic tree that have been discovered as a result of modern molecular data. Biologists strive to understand the evolutionary history and relationships of members of the animal kingdom, and all of life, for that matter. The study of phylogeny, the branching sequence of evolution, aims to determine the evolutionary relationships between phyla. Currently, most biologists divide the animal kingdom into 35 to 40 phyla. Scientists develop phylogenetic trees, which serve as hypotheses about which species have evolved from which ancestors. Recall that until recently, only morphological characteristics and the fossil record were used to determine phylogenetic relationships among animals. Scientific understanding of the distinctions and hierarchies between anatomical characteristics provided much of this knowledge. Used alone, however, this information can be misleading. Morphological characteristics, such as skin color, body shape, etc., may evolve multiple times and independently through evolutionary history. Analogous characteristics may appear similar between animals, but their underlying evolution may be very different. With the advancement of molecular technologies, modern phylogenetics is now informed by genetic and molecular analyses, in addition to traditional morphological and fossil data. With a growing understanding of genetics, the animal evolutionary tree has changed substantially and continues to change as new DNA and RNA analyses are performed on additional animal species. Constructing an animal phylogenetic tree. The current understanding of evolutionary relationships among animal or metazoa, phyla begins with the distinction between animals with true differentiated tissues, called eumetozoa, and animal phyla that do not have true differentiated tissues, such as the sponges, porifera, and the placozoa, figure 27. 6. Similarities between the feeding cells of sponges, choanocytes, and choanoflagellate protists, figure 27:12, have been used to suggest that metazoa evolved from a common ancestral organism that resembled the modern colonial choanoflagellates. The image on the left shows a choanoflagellate, which is a single-celled protist. The image on the right shows a sponge choanocyte cell that lines the inside of a sponge. The two cells appear identical. Both are egg-shaped with a cone at the back end. A flagellum juts out from the wide part of the cone. Figure 27. 12 choanoflagellates and choanocytes. Cells of the protist choanoflagellate clade closely resemble sponge choanocyte cells. Beating of choanocyte flagella draws water through the sponge so that nutrients can be extracted and waste removed. Eumetozoa are subdivided into radially symmetrical animals and bilaterally symmetrical animals, and are thus classified into the clades Bilateria and Radiata, respectively. As mentioned earlier, the cnidarians and ctenophores are animal phyla with true radial, biradial, or rotational symmetry. All other eumetozoa are members of the Bilateria clade. The bilaterally symmetrical animals are further divided into deuterostomes, including chordates and echinoderms, and two distinct clades of protostomes, including ecdysozoans and lophotrochozoans. Figure 27. 13a-b. Ecdysozoa includes nematodes and arthropods. They are so named for a commonly found characteristic among the group: the physiological process of exoskeletal molting followed by the stripping of the outer cuticular layer, called ecdysis. Lophotrochozoa is named for two structural features, each common to certain phyla within the clade. Some lophotrochozoan phyla are characterized by a larval stage called trochophore larvae, and other phyla are characterized by the presence of a feeding structure called a lophophore. Thus, the shorter term, Lophotrochozoa. Part A shows cockroaches. Part B shows phoronids, whose body is a slender stalk anchored to the ocean floor. Fine tentacles radiate from the top of the stalk. The tentacles and stalk resemble a flower. Figure 27. 13 ecdysozoa. Animals that molt their exoskeletons, such as these Madagascar hissing cockroaches, are in the clade Ecdysozoa. B. Phoronids are in the clade Lophotrochozoa. The tentacles are part of a feeding structure called a lophophore. Credit: a modification of work by Whitney Cranshaw, Colorado State University, Bugwood, org. Credit B. Modification of work by NOAA. Link to learning. Explore an interactive tree of life here. Zoom in and out and click to learn more about the organisms and their evolutionary relationships. Modern advances in phylogenetic understanding come from molecular analyses. The phylogenetic groupings are continually being debated and refined by evolutionary biologists. Each year, new evidence emerges that further alters the relationships described by a phylogenetic tree diagram. Link to learning. Watch the following video to learn how biologists use genetic data to determine relationships among organisms. Nucleic acid and protein analyses have greatly modified and refined the modern phylogenetic animal tree. These data come from a variety of molecular sources, such as mitochondrial DNA, nuclear DNA, ribosomal RNA, rRNA, and certain cellular proteins. Many evolutionary relationships in the modern tree have only recently been determined from the molecular evidence. For example, a previously classified group of animals called Lophophorates, which included brachiopods and bryozoans, were long thought to be primitive deuterostomes. Extensive molecular analysis using rRNA data found these animals are actually protostomes, more closely related to annelids and mollusks. This discovery allowed for the distinction of the protostome clade Lophotrochozoa. Molecular data have also shed light on some differences within the Lophotrochozoan group, and the placement of the Platyhelminthes is particularly problematic. Some scientists believe that the phyla Platyhelminthes and Rotifera should actually belong to their own clade of protostomes, termed Platyzoa. Molecular research similar to the discoveries that brought about the distinction of the Lophotrochozoan clade has also revealed a dramatic rearrangement of the relationships between mollusks, annelids, arthropods, and nematodes. And as a result, a new ecdysozoan clade was formed. Due to morphological similarities in their segmented body types, annelids and arthropods were once thought to be closely related. However, molecular evidence has revealed that arthropods are actually more closely related to nematodes, now comprising the ecdysozoan clade, and annelids are more closely related to mollusks, brachiopods, and other phyla in the Lophotrochozoan clade. These two clades now make up the protostomes. Another change to former phylogenetic groupings, because of modern molecular analyses, includes the emergence of an entirely new phylum of worm called Acolomorpha. These acyl flatworms were long thought to belong to the phylum Platyhelminthes because of their similar, flatworm morphology. However, molecular analyses revealed this to be a false relationship and originally suggested that acyls represented living species of some of the earliest divergent bilaterians. More recent research into the acolomorphs has called this hypothesis into question and suggested that the acyls are more closely related to deuterostomes. The placement of this new phylum remains disputed, but scientists agree that with sufficient molecular data, their true phylogeny will be determined. Another example of phylogenetic reorganization involves the identification of the Ctenophora as the basal clade of the animal kingdom. Ctenophora, or comb jellies, were once considered to be a sister group of the Cnidaria, and the sponges, Porifera, were placed as the basal animal group, sister to other animals. The presence of nerve and muscle cells in both the ctenophores and the Cnidaria, and their absence in the Porifera, strengthened this view of the relationships among simple animal forms. However, recent molecular analysis has shown that many of the genes that support neural development in other animals are absent from the ctenophore genome. The muscle cells are restricted to the mouth and tentacles and are derived from cells in the mesoglea. The mitochondrial genome of the ctenophores is small and lacks many genes found in other animal mitochondrial genomes. These features, plus the absence of Hox genes from the ctenophores, have been used to argue that the ctenophores should be considered basal or as a sister group of the Porifera, and that the evolution of specialized nerve and muscle tissue may have occurred more than once in the history of animal life. Although ctenophores have been shown as basal to other animals in the phylogeny presented in Chapter 27. 2. Debate on this issue is likely to continue as ctenophores are more closely studied. Changes to the phylogenetic tree can be difficult to track and understand, and are evidence of the process of science. Data and analytical methods play a significant role in the development of phylogenies. For this reason, because molecular analysis and reanalysis are not complete, we cannot necessarily dismiss a former phylogenetic tree as inaccurate. A recent reanalysis of molecular evidence by an international group of evolutionary biologists refuted the proposition that comb jellies are the phylogenetically oldest extant metazoan group. The study, which relied on more sophisticated methods of analyzing the original genetic data, reaffirms the traditional view that the sponges were indeed the first phylum to diverge from the common ancestor of metazoans. The ongoing discussion concerning the location of sponges and comb jellies on the animal family tree is an example of what drives science forward. Learning objectives. By the end of this section, you will be able to do the following. Describe the features that characterize the earliest animals and approximately when they appeared on Earth. Explain the significance of the Cambrian period for animal evolution and the changes in animal diversity that took place during that time. Describe some of the unresolved questions surrounding the Cambrian explosion. Discuss the implications of mass animal extinctions that have occurred in evolutionary history. Many questions regarding the origins and evolutionary history of the animal kingdom continue to be researched and debated, as new fossil and molecular evidence change prevailing theories. Some of these questions include the following. How long have animals existed on Earth? What were the earliest members of the animal kingdom, and what organism was their common ancestor? While animal diversity increased during the Cambrian period of the Paleozoic era, 530 million years ago, modern fossil evidence suggests that primitive animal species existed much earlier. Precambrian animal life. The time before the Cambrian period is known as the Ediacaran period. From about 635 million years ago to 543 million years ago, the final period of the late Proterozoic-Neoproterozoic era. Figure 27. 14. Ediacaran fossils were first found in the Ediacaran Hills of southern Australia. There are no living representatives of these species, which have left impressions that look like those of feathers or coins. Figure 27. 15. It is believed that early animal life, termed Ediacaran biota, evolved from protists at this time. Table describes eras in Earth's history associated with evolution of plant groups. In the first column, Earth's history is divided into 4 eons: the pre-Archean Hadean, Archean, Proterozoic, Phanerozoic. The oldest eon, the pre-Archean, spans the beginning of Earth's history to about 3.8 billion years ago. The Archean eon spans 2.5 to 3.8 billion years ago, and the Proterozoic spans 540 million to 2.5 billion years ago. The second column lists the eras within each eon. The Phanerozoic eon, from 540 million years ago to present time, is subdivided into the Paleozoic, Mesozoic, and Cenozoic eras. The third column lists the periods within each. The Paleozoic era, from 250 to 540 million years ago, is further divided into 7 periods. The Cambrian, from 500 to 540 million years ago. The Ordovician, from 435 to 500 million years ago. The Silurian, from 410 to 435 million years ago. The Devonian, from 319 to 410 million years ago. The Carboniferous, from 290 to 290 million years ago, and the Permian, from 250 to 290 million years ago. The Mesozoic era, from 66 to 250 million years ago, is divided into 3 periods: the Triassic, from 205 to 250 million years ago, the Jurassic, from 138 to 205 million years ago, and the Cretaceous, from 66 to 138 million years ago. The Cenozoic era, from 66 million years ago to modern times, is divided into 3 eras: the Paleogene, from 23 to 66 million years ago, Neogene, from 6 to 23 million years ago, and the Quaternary, from present to 2.8 million years. The last column indicates the time periods in millions of years. Illustration B shows geological periods in a spiral starting with the beginning of Earth's history at the bottom and ending with modern times at the top. The diversity and complexity of life increases toward the top of the spiral. Figure 27. 14. An evolutionary timeline. A. Earth's history is divided into eons, eras, and periods. Note that the Ediacaran period starts in the Proterozoic eon and ends at the start of the Cambrian period of the Phanerozoic eon. B. Stages on the geological timescale are represented as a spiral. Credit: modification of work by USGS. Most Ediacaran biota were just a few more cm long, but some of the feather-like forms could reach lengths of over a meter. Recently, there has been increasing scientific evidence suggesting that more varied and complex animal species lived during this time, and likely even before the Ediacaran period. Fossils believed to represent the oldest animals with hard body parts were recently discovered in South Australia. These sponge-like fossils, named Coronacalina acula, date back as far as 560 million years and are believed to show the existence of hard body parts and spicules that extended 20-40 cm from the thimble-shaped body, estimated about 5 cm long. Other fossils from the Ediacaran period are shown in Figure 27. 15a, b, C part A shows a fossil that resembles a wheel, with spokes radiating out from the center, imprinted on a rock. Part B shows a fossil that resembles a teardrop-shaped leaf, with grooves radiating out from a central rib. Part C shows a fossil that is much longer than it is wide, with many small ribs and a tail. Figure 27. 15 Ediacaran fauna. Fossils of A. Cyclomejusa, up to 20 cm, B. Dickinsonia, up to 14 m, and C. Sprigina, up to 5 cm, date to the Ediacaran period, 543-635 MYA. Credit: modification of work by Smith 609 /Wikimedia Commons. Another recent fossil discovery may represent the earliest animal species ever found. While the validity of this claim is still under investigation, these primitive fossils appear to be small, 1 centimeter long, sponge-like creatures, irregularly shaped and with internal tubes or canals. These ancient fossils from South Australia date back 650 million years, actually placing the putative animal before the Great Ice Age that marked the transition between the Cryogenian period and the Ediacaran period. Until this discovery, most scientists believed that there was no animal life prior to the Ediacaran period. Many scientists now believe that animals may in fact have evolved during the Cryogenian period, the Cambrian explosion of animal life. If the fossils of the Ediacaran and Cryogenian periods are enigmatic, those of the following Cambrian period are far less so, and include body forms similar to those living today. The Cambrian period, occurring between approximately 540 to 488 million years ago, marks the most rapid evolution of new animal phyla and animal diversity in Earth's history. The rapid diversification of animals that appeared during this period, including most of the animal phyla in existence today, is often referred to as the Cambrian explosion, figure 27. 16. Animals resembling echinoderms, mollusks, worms, arthropods, and chordates arose during this period. What may have been a top predator of this period was an arthropod-like creature named Anomalocaris, over a meter long, with compound eyes and spiky tentacles. Obviously, all these Cambrian animals already exhibited complex structures, so their ancestors must have existed much earlier. The illustration shows a seabed abundant with odd organisms, including tube-shaped worms anchored to the sea floor and animals that resemble cockroaches crawling along it. Swimming creatures somewhat resemble modern insects. Figure 27, 16 fauna of the Burgess Shale. An artist's rendition depicts some organisms from the Cambrian period. Anomalocaris is seen in the upper left quadrant of the picture. One of the most dominant species during the Cambrian period was the trilobite, an arthropod that was among the first animals to exhibit a sense of vision. Figure 27. 17a, b, c, d. Trilobites were somewhat similar to modern horseshoe crabs. Thousands of different species have been identified in fossil sediments of the Cambrian period. Not a single species survives today. Photos of B, C, D show 4 trilobite fossils. All are teardrop-shaped with a smooth, wide end. About 1/3 of the way down, the body is segmented into horizontal ridges. Figure 27, 17 trilobites. These fossils, a D, belong to trilobites, extinct arthropods that appeared in the early Cambrian period, 525 million years ago, and disappeared from the fossil record during a mass extinction at the end of the Permian period, about 250 million years ago. The cause of the Cambrian explosion is still debated, and in fact, it may be that a number of interacting causes ushered in this incredible explosion of animal diversity. For this reason, there are a number of hypotheses that attempt to answer this question. Environmental changes may have created a more suitable environment for animal life. Examples of these changes include rising atmospheric oxygen levels, figure 2718, and large increases in oceanic calcium concentrations that preceded the Cambrian period. Some scientists believe that an expansive, continental shelf with numerous shallow lagoons or pools provided the necessary living space for larger numbers of different types of animals to coexist. There is also support for hypotheses that argue that ecological relationships between species, such as changes in the food web, competition for food and space, and predator-prey relationships, were primed to promote a sudden massive coevolution of species. Yet other hypotheses claim genetic and developmental reasons for the Cambrian explosion. The morphological flexibility and complexity of animal development afforded by the evolution of Hox-controlled genes may have provided the necessary opportunities for increases in possible animal morphologies at the time of the Cambrian period. Hypotheses that attempt to explain why the Cambrian explosion happened must be able to provide valid reasons for the massive animal diversification, as well as explain why it happened when it did. There is evidence that both supports and refutes each of the hypotheses described above, and the answer may very well be a combination of these and other theories. The chart shows the percent oxygen by volume in the Earth's atmosphere. Until 625 million years ago, there was virtually no oxygen. Oxygen levels began to rapidly climb around this time and peaked around 275 million years ago at about 35%. Between 275 and 225 million years ago, oxygen levels dropped precipitously to about 15%, and then climbed again and dropped to the modern-day concentration of 22%. Figure 27, 18 atmospheric oxygen over time. The oxygen concentration in Earth's atmosphere rose sharply around 300 million years ago. However, unresolved questions about the animal diversification that took place during the Cambrian period remain. For example, we do not understand how the evolution of so many species occurred in such a short period of time. Was there really an explosion of life at this particular time? Some scientists question the validity of this idea, because there is increasing evidence to suggest that more animal life existed prior to the Cambrian period, and that other similar species, so-called explosions or radiations, occurred later in history as well. Furthermore, the vast diversification of animal species that appears to have begun during the Cambrian period continued well into the following Ordovician period. Despite some of these arguments, most scientists agree that the Cambrian period marked a time of impressively rapid animal evolution and diversification of body forms that is unmatched for any other time period. Link to learning. View an animation of what ocean life may have been like during the Cambrian explosion. Post-Cambrian evolution and mass extinctions. The periods that followed the Cambrian during the Paleozoic era are marked by further animal evolution and the emergence of many new orders, families, and species. As animal phyla continued to diversify, new species adapted to new ecological niches. During the Ordovician period, which followed the Cambrian period, plant life first appeared on land. This change allowed formerly aquatic animal species to invade land, feeding directly on plants or decaying vegetation. Continual changes in temperature and moisture throughout the remainder of the Paleozoic era, due to continental plate movements, encouraged the development of new adaptations to terrestrial existence in animals, such as limbed appendages in amphibians and epidermal scales in reptiles. Changes in the environment often create new niches, diversified living spaces, that invite rapid speciation and increased diversity. On the other hand, cataclysmic events, such as volcanic eruptions and meteor strikes that obliterate life, can result in devastating losses of diversity to some clades, yet provide new opportunities for others to fill in the gaps and speciate. Such periods of mass extinction, figure 27, 19, have occurred repeatedly in the evolutionary record of life, erasing some genetic lines while creating room for others to evolve into the empty niches left behind. The end of the Permian period and the Paleozoic era was marked by the largest mass extinction event in Earth's history, a loss of an estimated 95% of the extant species at that time. Some of the dominant phyla in the world's oceans, such as the trilobites, disappeared completely. On land, the disappearance of some dominant species of Permian reptiles made it possible for a new line of reptiles to emerge, the dinosaurs. The warm and stable climatic conditions of the ensuing Mesozoic era promoted an explosive diversification of dinosaurs into every conceivable niche in land, air, and water. Plants, too, radiated into new landscapes and empty niches, creating complex communities of producers and consumers, some of which became very large on the abundant food available. Another mass extinction event occurred at the end of the Cretaceous period, bringing the Mesozoic era to an end. Skies darkened and temperatures fell after a large meteor impact, and tons of volcanic ash ejected into the atmosphere blocked incoming sunlight. Plants died, herbivores and carnivores starved, and the dinosaurs ceded their dominance of the landscape to the more warm-blooded mammals. In the following Cenozoic era, mammals radiated into terrestrial and aquatic niches once occupied by dinosaurs, and birds, the warm-blooded direct descendants of one line of the ruling reptiles, became aerial specialists. The appearance and dominance of flowering plants in the Cretaceous era created new niches for pollinating insects, as well as for birds and mammals. Changes in animal species diversity during the late Cretaceous and early Cenozoic were also promoted by a dramatic shift in Earth's geography. as tectonic plates slid over the crust into their current positions, leaving some animal groups isolated on islands and continents, or separated by mountain ranges or inland seas from other competitors. Early in the Cenozoic, new ecosystems appeared, with the evolution of grasses and coral reefs leading to their wide distribution. Late in the Cenozoic, further extinctions followed by speciation occurred during ice ages that covered high latitudes with ice and then retreated, leaving new open spaces for colonization. Link to learning. Watch the following video to learn more about the mass extinctions. The chart shows percent extinction intensity of eliminated marine genera versus time in millions of years before present. Extinction intensity spikes at boundaries between periods, including the end of the Ordovician, which had 30% extinction occurrences of marine genera approximately 450 million years ago. The late Devonian had approximately 25% extinction occurrences of marine genera at roughly 375 million years ago. The end of the Permian had 50% extinction occurrences of marine genera approximately 250 million years ago. The end of the Triassic had roughly 30% extinction occurrences of marine genera 200 million years ago. And the end of the Cretaceous periods had over 30% extinction occurrences of marine genera roughly 70 million years ago. Figure 27, 19 extinctions. Mass extinctions have occurred repeatedly over geological time. Career connection. Paleontologist. Natural history museums contain the fossils of extinct animals, as well as information about how these animals evolved, lived, and died. Paleontologists are scientists who study prehistoric life. They use fossils to observe and explain how life evolved on Earth and how species interacted with each other and with the environment. The first paleontologists were scientists from other disciplines, such as geology, who evaluated the fossils they found to determine the concepts of extended ages of the Earth, prehistoric life, and extinction. They were aided in their work by surprise discoveries by laypeople and other more concentrated efforts by self-taught scientists. Mary Anning, for example, was a well-known fossil hunter who discovered, cataloged, and diagrammed the fossils of significant dinosaurs such as the Plesiosaurus. Scientists from throughout Europe consulted with her to deepen their own understanding and draw conclusions that laid the foundation for the discipline. Paleontology today relies on far more varied knowledge and technology than it did in Anning's day. A paleontologist needs to be knowledgeable in mathematics, biology, ecology, chemistry, geology, and many other scientific disciplines. A paleontologist's work may involve field studies, searching for and studying fossils. In addition to digging for and finding fossils, paleontologists also prepare fossils for further study and analysis. Although dinosaurs are probably the first animals that come to mind when thinking about ancient life, paleontologists study a variety of life forms, from plants, fungi, and invertebrates to the vertebrate fishes, amphibians, reptiles, birds, and mammals. Biophysics, biochemistry, geographic information systems, and data science are all additional fields of knowledge that paleontologists use to uncover the truth of the past. An undergraduate degree in earth science or biology is a good place to start toward the career path of becoming a paleontologist. Most often, a graduate degree is necessary. Additionally, work experience in a museum or in a paleontology lab is useful.